Hello, everyone. Thank you for joining this non-coding journal webinar entitled RNA Localization Regulation from Mechanism to Phenotype. My name is Madison. I'm the host today. So please be aware that this webinar is recording now and the record will be available to watch as early as possible on the webinar page of non-coding RNA journal. This webinar is organized by MDPI, a pioneer in scholarly open access publishing. MDPI has supported academic communities since 1996. Based in Basel, Switzerland, MDPI has the mission to foster open scientific exchange in all forms across all disciplines, and the supporting journal of this webinar is non-coding RNA, which is an international peer-reviewed open access journal on non-coding RNA research dealing with elucidating the structure, function, and biology of regularity non-coding RNAs. Non-coding RNA is published bi-monthly on-month by MDPI. Let me introduce our webinar chair of this. If you would be interested more about this webinar topic, we'll welcome you to read the relevant feature papers here. And if you would also be interested to submit the manuscripts on the top of this webinar, the non-coding RNA has also opened a special issue with the name of computational approaches for pinpointing the locations and the properties of non-coding RNAs, edited by the chair of this webinar, Professor Shizuka Uchida. And the submission deadline is, the submission deadline is the end of December 2023. You can scan the QR code for more information of this special issue. So please allow me to introduce the chair of this webinar, Professor Shizuka Uchida. He is a professor and co-director at the Center for RNA Medicine, Department of Clinical Medicine, Albert University. Professor Uchida's lab is interested in elucidating the functions of long non-coding RNAs and the epitranscriptomics enzymes using dry and wet lab techniques. He and his team have also built a number of bioinformatics tools to help analyze high throughput data. Professor Uchida utilizes knockout and transgenic mice to understand the effects of long non-coding RNAs and epitranscriptomics to cardiovascular system and development, as well as cardiovascular disease. And the keynote speaker today of this webinar is Dr. Gabriela Dombovic. She holds a PhD in biomedicine from the University Bombo Fala in Barcelona, specializing in the molecular characterization of microsatellites, repeat-derived long non-coding RNAs in colorectal cancer. After her PhD, she pursued his postdoctoral training in the Biofrontiers Institute, University of Colorado Boulder in the USA, working with John Rain on single molecule long non-coding RNA biology. Dr. Dombovic is currently leading an independent research group at Goethe University in Frankfurt, Germany, where she is continuing her work on the long encoding RNA biology, RNA localization, and its effects on RNA function and cellular phenotype using imaging, single cell, and biochemical technologies. So today we will only have one talk delivered by Dr. Dombovic, which will last for about 45 minutes, and the Q&A session will follow right after that. Our default Q&A time is 10 minutes, but if the discussion is intense, we will extend it according to the actual situation. If you have any further questions, please feel free to leave them in the Q&A box. So Professor Uchida, the floor is yours now, and I will stop sharing my screen. Feel free to start your presentation. Thank you.
Great. Thank you very much. Thank you very much for Gabby for allowing us to introduce you for this presentation. Localization of non-coding RNA is always an important topic. As we know, localizing nuclear may be involving or epigenetics or transcription regulation. Localizing cytoplasm may be involving RNA translational modification. So I'm very much interested in knowing what she can be shared. And also it's interesting to know that our current position is the same position that I held 10 years ago. So it's very interesting to talk and like to know what's going on at the our lab as well as the Professor Stephanie Demara's Institute as well. So it's your, please share your presentation. Thank you.
Perfect. So thank you for the introduction, and I would of course like to thank the organizers for inviting me to present today. And I would like to present you my previous as well as ongoing work on studying long non-coding RNAs. So my lab, as you've just heard, started very recently, so just last year. And in general, we are interested in studying long non-coding RNAs. So primarily, we are interested, of course, in understanding long non-coding RNA function, but we are more excited about knowing what are compartment-specific functions of RNA and which RNA elements can control RNA function, RNA localization, and then consequently RNA function. So just a very brief introduction. We all know now that the vast majority of the human genome is non-coding and importantly transcribed. But this is additionally being complicated by the fact that the majority of the non-coding genome is actually composed from repetitive DNA, which in itself is a very complex group of DNA elements. So repetitive DNA, based on the pattern of their distribution in the genome, can be divided either to interspersed repeats, which includes transposons and retrotransposons, for instance, or to tandem repeats, which includes ribosomal DNA and satellite DNA. And altogether, repetitive DNA has been shown to have important functions in the genome, such as, for instance, providing regulatory DNA elements, contributing to the evolution, but also giving rise to non-coding as well as coding RNAs, and in some cases also participating in genome organization. So collectively, all of these varying aspects of the non-coding genome were shown to be relevant, and also in some cases actually the driving force in the development of human diseases. And this is why we have this ongoing strong effort in trying to understand what is the function of these regions in both health as well as in disease. And one of the examples I would like to highlight here of how exactly complicated it can get is the development of facioscapulohumeral dystrophy, which is a muscle dystrophy where the muscles of the shoulder and the upper arm are progressively wasting. And this disease is the only genetic defect in this disease is reduction in the copy number of D4Z4 macrosatellites.
I'm sorry, Dr. Dombelik, your PowerPoint is frozen now in the first page.
Okay. So what you've missed, I will just briefly again summarize, is that in my lab, which started just recently, we are mostly interested in studying long non-coding RNAs, primarily, of course, understanding long non-coding RNA function, but also what are compartment-specific functions of RNA and which RNA elements control RNA function as well as RNA localization as well as function. So then I've talked about how the non-coding genome is additionally being, of course, complicated by the fact that the majority of the non-coding genome is composed from repetitive DNA, which based on the pattern of their distribution, we have two groups. And altogether, they have been shown to have important functions in the genome, from providing regulatory DNA elements, contributing to evolution, as well as giving rise to non-coding coding RNAs and participating in genome organization. And altogether, these varying aspects of the non-coding genome contribute to the development of human diseases, which is why, of course, we study these parts of the genome. And I believe that this is then where I can continue. One of the examples I would like to show you, which illustrates how complicated it can get, is the development of facioscapulohumeral dystrophy. And the only genetic defect in this disease is the reduction of the copy number of D4Z4 macrosatellite repeats. So in healthy individuals, these repeats are present in the subtelomeric region of chromosome 4, and there are between 11 to 150 copies. But in patients from FSHD, basically what happens is that we have a reduction of these repeats, but at least one repeat unit is necessary for the development of the disease. And in healthy individuals, these repeats act as a platform for polycomb group proteins, which then results in a repressive chromatin structure and the repression of the adjacent genes, which are thought to have the role in the phenotype of the disease. And in the patients, what happens when these repeats are, there is less number of those repeats, is that the chromatin structure is loosened, which allows for the expression of a long non-coding RNA from these repeats, which in turn recruit triturax group protein H1, which further activates local transcription and the expression of adjacent genes. So this was just one example how complicated things really can be. And in today's presentation, I would like to, it's divided in two parts. And in the first part, I would like to talk about my previous work on repeats and long non-coding RNAs. And then in the second part, on the role of splicing in the regulation of RNA localization and function. During my PhD, we were focusing on how epigenetic changes can affect the non-coding genome landscape in cancer. And in particular, we were focused on colorectal cancer, where we knew that global DNA hypomethylation as well as local DNA hypermethylation occur in the majority of colorectal cancers. So while in normal colon epithelium, the promoter regions of tumor suppressor genes, they are normally hypomethylated, resulting in their transcriptional activation, the pericentromeric and centromeric regions, which are normally very rich in DNA repeats, are highly methylated, and this state has been linked to genomic stability. On the other hand, in colon cancer, what happens is that the promoter regions of tumor suppressor genes get hypermethylated, resulting in their transcriptional inactivation, while pericentromeric and centromeric regions get hypomethylated. And we didn't really know in detail what are the consequences of this DNA hypomethylation, but we knew that this state has been linked to increased genomic instability. And one of the reasons why studying these consequences was difficult, because for the most part, and especially before T2T project, DNA repeats were not assembled in the human genome. So even if we found certain sequences affected, we really wouldn't know where to look for the consequences of these changes. And this is somehow how my PhD started, where we found that a primate-specific macrosatellite repeat called MBL2 is frequently hypomethylated in colon cancer. But these repeats, they were not assembled in the human genome, so we only had just a snapshot of the repeat array and a bit of upstream and downstream regions, but we didn't know where within the genome they are. But what we did know is that each repeat unit has 1.4 kilobases, and it is very highly GC rich. So we found it hypomethylated in colon cancer, but it has also been found hypomethylated in other cancer types. And as you can see here, it is very frequently hypomethylated in neuroblastoma as well as hepatocellular carcinoma. So we really wanted to know what happens then when these repeats get hypomethylated. So since we first didn't know where they are localized, we started by mapping which chromosomes contain these macrosatellites. And here we took advantage of human rodent monosomatic somatic cell hybrids, which contain one human chromosome in the rodent background. And because this is a human primate-specific sequence, it is not present in the rodent background. Therefore, we can use it as a perfect mapping panel for knowing which chromosomes have the sequence. So we did this by doing just a regular PCR, employing different primer sets across the repeat unit. And what we could see is that chromosomes 9, 13, 14, 15, 21, as well as Y, have the presence of the entire repeat unit, while different fragments of this repeat could also be amplified on other chromosomes. So then we wanted to know where within the genome these repeats are localized. And for this, we did DNA fish on metaphase spreads. And what we could see is that repeat arrays of MBL2 macrosatellites are localized in the short arms of acrocentric chromosomes 13, 14, 15, and 21 in the pericentromeric region. So then we wanted to know where within the 3D genome they're placed. And for this purpose, we employed 3D DNA fish coupled with nucleophosphine immunofluorescence, which labels nucleoli. And we could very nicely see that these repeats are localized in the perinucleolar region. So then we went on looking what happens when these repeats are hypomethylated. And I'm sorry that you have to see everything all at once. I would normally guide you through the data, but I will still try to do that. So first, we looked at the expression. So this was, of course, the most obvious. Although I have to say at that time, it was not the most obvious thing because expression of repeats was not something that people really considered in the time. But we looked at whether there is any indication that there is expression of those repeats when they're hypomethylated. And here we use cell lines that have either endogenously hypomethylated MBL2 repeats or they have them highly methylated. And indeed, we could see that there is a higher expression in the cell line that has them hypomethylated. But importantly, we could induce this expression in the cell lines that have them highly methylated by hypomethylating the DNA with 5-aza-2-deoxycytidine, which is an epigenetic drug that can inhibit DNA methylation. And you can see here that once we inhibit DNA methylation, there is an increase in expression. So normally pericentromeric heterochromatin is not being maintained only by DNA methylation, but we also have other factors such as, for instance, histone deacetylation. So we wanted to see whether histone deacetylation might have any function in the transcriptional regulation of these repeats. And for this, we inhibited histone deacetylation by another epigenetic drug called TSA. And we did that either if we previously hypomethylated DNA with AZA or on top of highly methylated DNA. And what you can see here is that when we hypomethylate, there is an induction of expression, but this induction is much higher if on top of previously hypomethylated DNA, then we further inhibit histone deacetylation. And as a proof of concept, here we also used HCT116 double knockout cell line, which has DNMT1 and DNMT3B knocked out, and therefore these repeats are completely unmethylated. And here what we can see that if we add AZA, there is no induction of expression, which then excludes the possibility that this could be due to any non-specific effects. If we add TSA alone here, there is tens of thousands of times higher expression of this repeat compared to the untreated cells. So all of this together shows us that both DNA methylation and histone deacetylation regulate the expression of these macrosatellites, but the DNA methylation is the ceiling mark in their transcriptional repression. Could you please just let me know if you at least see my mouse, so that you see where I'm pointing?
Yeah, we can see the mouse.
Oh, perfect. So then, thanks. So then we wanted to go back to the primary patients and see if this happens in the primary patients. And here you can see the bisulfite sequencing analysis, where in gray I'm showing the normal tissue, here the tumor tissues. And these repeats, as I've said before, they're normally highly methylated. But in patients that have them highly hypomethylated, there is also an induction of expression. And this was not a very frequent event. It occurred in approximately 8% of colorectal cancer patients. And we have accordingly designated this transcript TMBL for tumor-associated MBL2 transcript. So then, of course, we wanted to know about more about this RNA. So where is it localized? Is it a stable RNA? Does it have a defined length? Because, of course, it could also be just a run-through transcript, especially considering that we really didn't know the context within which it's expressed. So by fractionation, what we could see is that this RNA is enriched in the chromatin. And that if we inhibit transcription by actinomycin D and then analyze RNA stability, we can see that this is a highly stable transcript comparable to that of MALAT1 or protein coding RNAs. And by Northern blot on cell lines, when we induce expression of this RNA by AZA and AZA-TSA, we can see that there are several transcripts being made of high molecular weight, approximately 10 and 13 kb, and also a smaller one, although fainter, around 4.2. So altogether, this shows that DNA methylation leads to expression of a chromatin-associated non-coding RNA, which is very stable and also very long. So as I told you before, we didn't really know where this RNA comes from, because there were multiple chromosomes that had this repeat map. So then we performed qPCR and northern blot on somatic cell hybrids after inducing the expression with the ASA and ASA-TSA. And we could see that this RNA comes only from chromosomes, acrocentric chromosomes 13, 14, 15, as well as 21. By single molecule RNA imaging, we could see that this RNA is exclusively nuclear, where it is widely distributed. As you can see here, there are many diffused signals, but it preferentially actually forms these aggregates or clusters that have high density of TMBL RNA. And by state super resolution microscopy, we could actually really see that these aggregates are composed of hundreds of independent TMBL molecules. And this was indirectly validated also if we monitor what happens with this RNA during mitosis, where in early prophase, these aggregates start to disassemble, and then hundreds of independent RNA molecules are released in the cytosol. So because we knew that the DNA was in the perinucleolar region, we also looked at where are these aggregates, and as well they were placed here, you can see by RNA imaging, and again, nucleophosphine immunofluorescence. They are localized in the perinucleolar region. So then we looked at what other bodies exist in the perinucleolar region, and there were two: one perinucleolar body as well as some 68 perinucleolar body. And what then we co-stained with some 68 together with TMBL RNA fish, and what we could see is that these two bodies coexist together. They form specifically under hypomethylation and in cancer context. But what was intriguing is that they never protruded within each other. They're always right next to each other, and they even seem to be making mirroring structures, implying that they either form around or they depend on the same chromatin structure. So with this, I would like to summarize this part. And what I've showed you is that MBL2 macrosatellite repeat arrays are localized in the short arms of acrocentric chromosomes 13, 14, 15, and 21 in the pericentromeric regions, where in normal conditions, they display features of constitutive heterochromatin with high levels of DNA methylation and K9 trimethylation, and they're also transcriptionally inactive. Upon cancer-specific hypomethylation, we can see that there is an expression and generation of a long non-coding RNA, which is further enhanced to a much greater extent if we then acetylate the histones. And this RNA is exclusively nuclear, where it is widely distributed, but it preferentially forms these perinucleolar aggregates or structures, which are adjacent to some 68 body. So now I would like to switch to the second part of my talk and talk about our recent work on studying RNA localization, and specifically the role of splicing, you will see intron retention, in the regulation of RNA localization as well as function. So we know now that long non-coding RNAs can occupy virtually all cellular compartments, both in the nucleus as well as in the cytoplasm. And while in the nucleus, long non-coding RNAs, they can regulate the expression of proximal genes by bringing either certain transcription factors or chromatin remodelers to the proximal genes. They can also mediate transchromosomal interactions and regulate the expression of distal genes. In both the nucleus as well as in the cytoplasm, long non-coding RNAs can act as scaffolds, and in post-transcriptional regulation. And for instance, some of the most famous examples of scaffolding include the nuclear bodies that I've just shown you. These include also speckles, paraspeckles, as well as, for instance, stress granules in the cytoplasm. So long non-coding RNA loci are also very complicated in the mechanism by which they function. So they can function either via RNA-based mechanisms, which include bona fide long non-coding RNA such as exist and telomerase, But also it has been shown that many encode for unannotated open reading frames, which can give rise to peptides, and many also seem to be bifunctional. Also, many long non-coding RNA loci act as DNA elements to regulate the expression of nearby genes, where in some cases the non-coding product has a function, while in some cases it seems to be completely dismissible. And lastly, it seems that some also don't have any apparent function. So one of the key features that can distinguish a protein coding RNA and a long non-coding RNA is subcellular localization, where long non-coding RNAs are much more abundant in the nucleus, specifically in the chromatin of the cell, while of course protein coding RNA are more abundant in the cytoplasm for translation. And another key feature that can distinguish them is splicing or intron retention, which in general is much higher in long non-coding RNAs, as you can see here. And also interestingly, it's very much cell type specific. So intron retention occurs once a pre-mRNA has been fully processed, but one or more introns have been left in the mature transcript. So this RNA is capped, polyadenylated, other introns are spliced out, but then there are just some introns that are left in the transcript, and normally it will result in nuclear retention of the RNA. So the importance of this process, the one that has been demonstrated but also speculated, is in stress response and in disease, giving rise to novel non-coding RNAs, and also in gene regulation. But we still have many challenges and opportunities in studying intron retention, specifically in defining specific role of intron retention, as well as mechanisms that regulate it, and also providing novel insights into their function in physiology as well as disease, and also potentially exploring it as a therapeutic vulnerability. So this is something that I'm very interested about. So what we actually want to understand is how intron retention modulates RNA function in a way where these long intronic sequences can provide compartment-specific binding regions, for instance, for certain proteins or chromatin regions, and as such contribute to diversification of the RNA function compared to the spliced cytoplasmic counterparts, or they can just spatially separate the RNA in a different compartment. As well as we know that this process is highly modulable by external stimuli as well as in disease. So another thing that we want to understand is how disease contexts and stress affect this process. How does this in turn affect RNA localization, RNA function, and then ultimately the cellular phenotype? And today I would like to show you two such examples from our recent work where we showed that intron retention actually has a very important role in regulating their function. So first one is TAG1, which is a very interesting non-coding RNA for several reasons. So this is actually one of the most conserved long non-coding RNAs between human and mouse. And on the subcellular level, it is localized in both the nucleus as well as in the cytoplasm. And again, this property is conserved between human and mouse. Recently, we showed that there is a region in the 5' region of TAG1, which has high phylogenetic conservation score, which could potentially indicate that this isoform of TAG1 could be protein coding. So the mouse model for TAG1 was generated many years ago in the RIN lab, where what they did is that they removed the TAG1 locus, but left the promoter as well as act of transcription intact. And in a follow-up study, what we could see is that the deletion of this locus actually leads to complete infertility in male mice, as you can see here. And that this is due to those mice having both low sperm count, but also abnormal sperm morphology. On the expression level, we wanted to, of course, also know what happens to gene expression, because TAG1 is relatively high expressed across multiple different tissue types. So then we performed RNA sequencing and compared wild-type and knockouts across different tissues. And indeed, we could see that there are many genes that are affected, being either upregulated or downregulated. But what was quite intriguing is that there were several pathways which are affected in a general, so across different tissue types. implying that TAG1 might have a general function, but also there were several pathways which were affected in a tissue-specific way. So we wanted to use these changes as a platform to understand which are the functions of each of the putative regulatory elements present in the TAG1 locus on the regulation of expression of those genes. So first we wanted to understand whether TAG1 might have any regulatory DNA elements. And here it is important to keep in mind that the majority of long non-coding RNA loci can act DNA elements regulate the expression of nearby genes by serving as a platform for either certain epigenetic modifications or transcription factors that can directly affect the expression of nearby genes. And consequently, if we remove this locus, then the expression of those genes will be affected, and whether they are going to be upregulated or downregulated depends on the initial nature of the regulation of the locus itself. So with this in mind, we analyzed differential gene expression 1 megabase upstream as well as downstream of the TAG1 locus in testis, and TAG1 is shown here in the middle. And as you can see, there was a subset of genes highlighted here in red that was upregulated once we removed the locus. So this was indicative that TAG1 locus actually might have a cis repressory function. And to see this in more detail, we performed the same analysis across all the other tissues for which we had the RNA-seq for, and we could see that the majority of those genes is upregulated once we remove the locus, irrespective of the tissue type. And as a further demonstration, we generated a hybrid mouse model by using two strains of mice, one that has wild type and the other one that has TAG1 knockout. And because these mice have certain SNPs, this allows us then to map the gene expression to the allele that either has TAG1 knocked out or has the wild type allele. And here what we could see is that indeed these genes, they are upregulated on the allele that has TAG1 knocked out, while the allele that has TAG1 present, these genes are not affected. So this altogether shows us that TAG1 DNA or the locus has regulatory elements that can affect the expression of downstream genes, and it is supportive of a cis repressory role for the locus. So next, we wanted to understand whether there are any purely RNA-based functions. And for this, we generated a rescue mouse model where a doxycycline-inducible TAG1 transgene was introduced in the knockout background. And in choosing the transgene here, we chose an isoform that lacks the 5' region that is protein coding, ensuring that we would be monitoring only the non-coding RNA effects. And once we generated the rescue mouse model, then we harvested the testis and analyzed, first of course, the level of rescue of TAG1 and then gene expression changes. But unfortunately, what we could see is that the rescue was not complete. So we could achieve only approximately 5% of the wild type levels, which of course was very disappointing at first. And there was no rescue in the phenotype of the mice. But we were nevertheless pleasantly surprised to see that there is a subset of genes which is responsive to even these lower levels of TAG1 RNA. So here in red, you can see genes that are upregulated once we remove the locus, and they are reciprocally regulated once we introduce TAG1 RNA even at low molecular count. And just of note, these genes that were affected in cis, they are not affected by introducing this lower quantity of TAG1 RNA. So this shows us that TAG1 RNA has biological activity in trans, even at a lower molecular count. And lastly, we also wanted to see whether TAG1 might actually have any protein-based function. If you remember in the beginning, I've showed you that there is a region with high phylogenetic conservation score, which could be implying that there is also some protein coding function to the RNA. So initially we started by screening, so we took a very unbiased approach and we screened for conserved open reading frames across human and mice. And there was just one open reading frame highlighted here in red that had very high levels of amino acid conservation. So of course we wanted to know then whether this ORF can get translated. And as a first strategy, of course, we analyzed multiple, both human as well as mouse ribosome profiling data, and all of them show the same. As you can see here, there is high ribosome profiling coverage in the 5' region of TAG1. which spans through the entire length of open reading frame 1. And then there is a sharp decrease at its stop codon, which is indicative of ribosome release upon a translation termination. And afterwards, there is no coverage above the background level. Furthermore, in collaboration with Sebastian van Hees, we performed in vitro translation assays using long non-coding RNA tag 1. And what we could see is that there is just one protein product being made, the size of which corresponds to the predicted size of open reading frame 1. So this altogether points towards this being a bona fide open reading frame. So then we wanted to know where this peptide is localized within the cells. So for this, we performed, again, I have to apologize that you're seeing everything at once. So, because otherwise I would guide you, but I hope that still you can follow my pointer. So once we overexpress tag1 peptide, we can see that it localizes in both the nucleus as well as in the cytoplasm. But while in the nucleus, it has this wide distribution, in the cytoplasm, it co-localizes exclusively with the mitochondria. But actually, not only does it co-localize with the mitochondria, it also affects mitochondrial functionality, which we assess by using chloromethylexrosamine. So this is a lipophilic dye, which accumulates in the negatively charged interior of mitochondria. Therefore, we can stain the mitochondria using this dye. And then if we don't see any staining with this dye, this somehow implies that the mitochondria have been compromised. So here, if we overexpress tag1 RNA that lacks open reading frame 1, showed here in white, the staining with chloromethylexrosamine is intact across all cells. But if we overexpress either human or mouse tag1 protein, we can see that the majority of cells actually lack staining with chloromethylexrosamine, which indicates that this protein impairs mitochondrial functionality. So just to summarize this part, what I've showed you is deleting what is still considered a non-coding RNA locus leads to complete infertility in male mice. But on the molecular level, that there are multiple modalities which are present in the tag1 locus, one being at the level of the DNA, one being at the level of the RNA, where the RNA itself can regulate the expression of genes in trans. And lastly, that tag1 also harbors a small open reading frame whose encoded product is implicated in mitochondrial maintenance. So of course, now it will be interesting to see which of these, or maybe their combination, are responsible for the infertility phenotype. But something that I was very intrigued by and also very passionate about trying to answer is how tag1 RNA manages to have these dual functionalities. And this, again, brought me back to tag1 localization, because in the end, we know that RNA localization is key for its function. And as I've showed you before, tag1 is localized in both the nucleus as well as in the cytoplasm. So this was an indication that undoubtedly this dual localization gives the opportunity to this RNA to perform these multiple functions. So we wanted to understand how tag1 maintains this dual localization. And long story short, we looked at multiple different aspects, but what we could see by RNA sequencing is that across both of tag1 introns, so tag1 has 2 introns, there is high RNA-seq coverage, which is not normally seen for an efficiently spliced intron. So this already pointed that maybe there is something happening with those introns, and then we employed single molecular RNA imaging targeting either exons or intron 1 as well as intron 2. And what we could see, regardless of the cell type, so we analyzed really a lot of cell lines and all of them show the same, which is that tag1 is localized in both the nucleus as well as in the cytoplasm, but nuclear tag1 retains both of its introns. And also what is important to note is that there is a positive correlation between overall nuclear enrichment of tag1 RNA and intron retention, which implies that intron retention might be dictating overall nuclear cytoplasmic balance of tag1 RNA and consequently its molecular function. So we know that long non-coding RNAs are in general not efficiently spliced. So we thought that maybe this is just a consequence of slow splicing kinetics and eventually tag1 will get spliced. So we analyzed the stability, splicing, and localization of tag1 RNA once we inhibited transcription with actinomycin D. And here, of course, we did that by single molecular RNA imaging. And what we could see is that nuclear TAG1 is actually much more stable compared to cytoplasmic TAG1, which gradually increases over several hours. But importantly, that nuclear TAG1 stays in the nucleus and it stably retains both intron 1 as well as intron 2. So this shows us that this is actually a stable, long-lived RNA. So this is where we started to think whether maybe somehow we could use this intron retention in our favor and try to modify the localization of TAG1 RNA by modulating the levels of intron retention. And here we teamed up with Marvin Karuters at BioFrontiers, who developed a novel antisense oligonucleotide chemistry called thiomorpholino oligonucleotides, which work extremely efficiently. So these antisense oligonucleotides have higher affinity to the RNA, and they are also resistant to exonucleases, which makes them perfect for splicing studies. So we tested several different regions against which we would place these antisense oligonucleotides, but this was the region that actually worked the best. So 2 nucleotides in the exon and 18 nucleotides in the intron of both donor splice sites. And then if we treat the cells with these antisense oligonucleotides, we can see that there is a higher and a quite efficient retention of both intron 1 as well as intron 2. And on the subcellular level, what happened is that we managed to change the localization of the RNA from being both nuclear as well as cytoplasmic to being predominantly nuclear, which was due to this higher retention of both intron 1 as well as intron 2. So this further demonstrates that intron retention is the driving force in nuclear retention of TAG1 RNA, but also it allows us to try to test different antisense oligonucleotides technologies to target RNA localization. And what we noticed that once we actually change this localization of the RNA, over time, the cell viability is reduced. That also further confirms that the RNA really cares, the cell really cares where the RNA is localized. So in summary, here what I've showed you is that TAG1 requires splicing, or in other words, intron retention, to maintain subcellular distribution as well as stability, that where TAG1 is expressed within the cell is important for the cell, and that we can use these antisense oligonucleotides to modulate RNA localization, which also has a good therapeutic potential. And this brings me to the last part of my talk, and I would like to slightly switch gears here and talk about protein coding RNA. TERT, which encodes for the catalytic subunit of the telomerase complex. So we know that this complex is extremely important because it extends the ends of our chromosomes, and it's one of those complexes without which we wouldn't be able to exist. And important also to note is that TERT is the limiting component in the assembly of the telomerase complex. So due to its biological activity, telomerase is expressed in frequently dividing cells, such as, for instance, in fetal development and in stem cells, while on the other hand, in most body cells, telomerase is silent, which leads to progressive shortening of telomeres with each cell division. And once a critically short length is reached, so-called the Hayflick's limit, the cell undergoes senescence. On the other hand, telomerase is reactivated in the majority of cancers, which then gives these cells this capacity to proliferate indefinitely. So this project, again, initially started as a collaboration with the lab of Tom Czech, because due to the nature of TERT expression, TERT is a very lowly expressed RNA, so we cannot really efficiently detect by immunofluorescence which cells express TERT. So we wanted to use an alternative strategy and apply RNA imaging to visualize which cells has TERT expression and which cell doesn't. And this actually led us to a very unexpected finding where we designed the probes against only the exons of TERT, and we could detect that the vast majority of TERT RNA is localized in the nucleus. And again, I'm repeating, this is a protein coding RNA that needs to be exported to the cytoplasm for translation. And as you can see here, there is only 2 to 3 molecules in the cytoplasm which are available for translation. So this was indicated that there might be an RNA-based mechanism that regulates the availability of third RNA. So we wanted to explore how does this work? So by which mechanism is TERT being regulated and retained in the nucleus? And coincidentally, this also led us to intron retention. So TERT has a total of 15 introns, and we can see that all of them are efficiently spliced, except for 2 introns, intron 11 as well as 14, where we could already observe that there is higher RNA-seq coverage compared to other efficiently spliced introns. And then we use single molecular RNA imaging, where we either targeted exons of TERT or intron 11 and 14, and we also used intron 2 as a control for an efficiently spliced intron. And as you can see here, nuclear TERT, so 100% retains intron 11. It also retains intron 14, although to a lesser extent. And we can see intron 2 forming 1 to 2 punctuate dots in the nucleus, which is what we would expect, marking active transcription sites. So we again wondered here whether this is just a transitory state, because this is a protein coding RNA, and maybe these introns need to be post-transcriptionally spliced, and it just takes a bit more time. So we again inhibited transcription using actinomycin D, and then used single molecular RNA imaging to assess the localization, splicing, and the stability of TERT RNA. And what we could see is that cytoplasmic TERT is actually much less stable than nuclear TERT, and it goes down very quickly, although there was not a lot even to begin with. But importantly, nuclear TERT is very stable, and it also stably retains intron 11 as well as intron 14, which shows that these are actually stable and long-lived transcripts. Next, we also looked at the conservation, and each species that we looked at had retention of at least 1 intron within TERT. So TERT intron retention is conserved, but the identity of which intron is conserved varies between species. So this somehow tells us that the cell knows how to put evolutionary pressure on having intron retention in TERT, but there is not an evolutionary pressure on a specific sequence. So here then we wanted to use these antisense oligonucleotides as a proof of concept to see whether we can also retain a protein coding RNA in the nucleus. So we designed them against exon 11, intron 11, and indeed we were successful in achieving retention of those of the intron and the retention of the RNA in the nucleus. And then we looked at typical consequences that normally occur once we don't have TERT available. And indeed, we had a reduction in cell viability after several days, cell cycle arrest in G2M phase, as well as an increase in genomic damage. And just to really show that this has something to, that this is due to TERT, we performed a rescue experiment where we co-expressed TERT thymorpholinos to go together with spliced TERT RNA, which is therefore not a target for these oligos. And indeed, we could rescue the effect in cell cycle, cell viability, as well as in genomic damage. So just to summarize this part, what I've showed you is that TERT is actually a predominantly nuclear RNA, and that this is due to stable retention of intron 11 as well as 14. That of course, splicing and overall nuclear cytoplasmic balance of TERT RNA is important for the cell. And indeed, we can use these antisense oligonucleotides to modulate RNA localization. So with this, I would like to finish. And I hope that today I managed to show you and to convince you that there are multiple hidden layers of complexity within the non-coding genome, and that our knowledge of how non-coding genome and what non-coding genome does is continuously growing. And this is, of course, complicated by the fact that there are multiple functionalities, on top of which we also have to sum up potential splicing effects and specialized localization, which will all have a very profound effect on how RNA works. And it is our task to try to understand this as better as we can. So with this, I would like to, of course, thank you for your attention. I would like to thank, of course, my current team. And today I have showed you my previous work, mostly. So, of course, I would very much like to thank the members of the RIN lab, also Perucho and Forcales labs, our previous collaborators as well as current ongoing collaborations. And with this, I am happy to take any questions.
Thank you very much for a nice presentation. Let me start off the questions. You have shown very nice pictures, but how difficult is it to design probes? For example, like ncRNA 1 in 3 works and RNA gap mirrors 1 in 10 works. How about how many trial that works for your RNA imaging?
So designing probes for RNA imaging, if I understood you.
Yes.
So normally, if the transcript is long enough that we can play, I don't know.
I think you got frozen.
So normally, so up to now, every transcript that we try to detect with RNA fish, we didn't have any problems. The problems would occur when the region would be maybe small. So let's say that we could place only 8 probes, and then the signal was not strong enough to see anything. So I would say that as long as the RNA is long enough and we can place maybe more than 15 probes, then it should work. And of course, if the RNA is expressed.
How do you confirm the specificity of your probes?
So there are different ways. So for instance, for the MBL2 repeats, what I was doing was downregulating the RNA with gap mirrors, which are specific to this repeat. And then if there was no transcript detected, which was in this case, then this was showing the specificity. Also, normally we use a cell line that doesn't express the gene of interest. Sometimes we can also do cross-species validation, but always normally we have to have some control that this is a specific process.
There are 3 questions in Q&A. Can you open the Q&A? I mean, I can read it as well, but it might be easier. So
no.
Can you open the Q&A?
Yes.
Yeah, okay. So the first question is, are the cell lines named here hypermethylated?
So I'm reading, so whether we know anything about RNA methylation?
Yeah, the cell lines they have used. RNA cap PC3 E006AA.
Sorry, could you just please repeat, because I was reading the questions in the Q&A.
The Q&A, the first one is, are the cell lines that you have used hypermethylated? Hypermethylated, are they those cells?
Ah, okay, I see now the questions.
Yeah.
So I didn't use those cell lines. So this is PC3, and I didn't use this cell line, so I wouldn't know.
Okay. Then let's go to the next question by Prabhu.
So RNA modifications, so yeah, I think that there is some evidence that RNA modification, specifically methylation, can affect splicing. And of course, then this could potentially affect the localization of the RNA. This is something that we are currently looking into, but I don't have firsthand data yet to tell you if this is the case with TERT or TAG1.
Okay, there's a third question is from K-scale. Switching long non-coding function due to the presence of introns.
Yes, I actually, so that's a very complicated question, because there are many mRNAs that also retain specific introns, and normally it is thought that maybe they're just being quarantined in the nucleus, maybe in speckles or paraspeckles, waiting for a stimuli when they are needed, and then their splicing is triggered and they're going to be exported to the cytoplasm. And there is very little evidence, but that's maybe because we haven't really looked at that in much detail, that these RNAs could also function as long non-coding RNAs. And of course, there's a lot of work, because there's hundreds of transcripts that could be doing that. This is something we are interested in studying currently in the lab. I believe that yes, but let's see.
Then there is a question. balance between tag1 and non-coding RNA and peptide?
So we don't know what regulates the balance. Potentially could be intron retention. So if there is more intron retention, we have more nuclear RNA, and then we have less cytoplasmic and less peptide. The way that the peptide would be targeted to the mitochondria, so we analyze whether there is an existence of certain signal peptides or mitochondrial targeting sequences. And the peptide actually has a conserved, the protein has a conserved signal peptide in the N terminus, and the C terminus actually has hydrophobic transmembrane domain, which could explain targeting to the mitochondria.
Okay, add on to this question, given that tag1 is also expressed in female, yet the knockout mouse that you have seen, you only see the phenotype in male.
Yes.
And then the mechanism you're coming down to potentially non-coding RNA function, or could also be the peptide that they encode. Could these peptides be translated in female as well as in male?
Yes, but we don't say that it's the peptide causing the infertility.
Okay.
So we actually know that this infertility occurs once we delete the locus, but we don't know if it's because of the RNA, the protein, or the DNA. So
yeah, given that situation, both, like, I guess DNA, RNA, and protein or peptide of tag1 is there for male and female, yet you only see the phenotype in male. Then potential phenotype, the mechanism that you came down to is the mitochondrial function, which is also present in male and female. I'm asking that a little bit more
yes,
critical.
so we are not saying that
How
this
did you explain that? Yeah,
but
sex
in
difference.
general, the way that male and female generation of sperms or oocytes works is very much different. So the process that is affected very likely, because we see a reduction in sperm, the process that we see would be during generation of sperm. And one of the things that we know about tag1 is that its expression is being dynamically changed during spermiation. But again, from here, we still cannot conclude whether it's the RNA or DNA or the potential protein that affects this.
Okay. And there's another question.
Yeah, I'm trying also to read.
Two more. Yeah, sorry, this one was answered. Well, there's more. Evgenia try to enhance splicing with CS effects.
No, we haven't. That's actually a very good idea. Yeah, that would be very interesting to see. We haven't done it, but good suggestion.
And additional second question is dual localization, long non-coding RNA, are general more enriched in short.
Yes, so I think that here, so what has been shown, and actually this was a very comprehensive studies from Sebastian Van Hesch. So this was published in Cell 2019. So they have actually analyzed the translational landscape of the human heart, and what they found is that there are many hundreds of annotated long non-coding RNAs that actually have demonstrated long non-coding RNA functions that also produce a protein. So this seems to be a much more common phenomena than what we initially thought.
Okay, there's another question. Mutation, whether they produce desired protein or peptide.
I'm not sure that I understand the question.
So I'm assuming they're asking more towards whether the mutation changes.
So I would assume that if we were able to mutate to achieve enhanced export of the RNA, that would likely consequently also increase the quantity of the protein. So we have currently done this by trying to retain the RNA in the nucleus, but currently in the lab, we are also trying to determine if we can do it the other way around. So we are specifically trying to understand what sequences and proteins regulate this synchron retention, and then whether we can somehow, either by antisense oligonucleotides, as you mentioned, by endogenous mutation, affect those sequences and then achieve a greater export and then potentially more protein.
When you study TAG1, are you concerned about, if you are studying human, for example, are you concerned about the sex of the cell, the donor cells?
So in cell line models, no.
Yes.
No. But we did look at whether there are any so we of course had the same thing that you're thinking. So we also looked if there's any difference. For instance, if we look at female mouse embryonic stem cells or male, but here we didn't see any differences in the localization of the RNA.
I guess that's all the questions that we had, and I think we're 5 minutes past the time. And thank you very much for a nice presentation. I certainly did enjoy it. Thank you very much for your time. And thank you very much for all the attendance. I hope you enjoy Gabby's talk, and have a nice day or night or morning, and hope to see you next time.
Thank you. Bye.
Bye.